Loosely attached to the cytoplasmic side of the membrane, the G protein functions as a molecular switch that is either on or off. When GDP is bound to the G protein is inactive. When the signaling molecule binds to the extracellular side of the receptor is activated and changes shape. Its cytoplasmic side then binds to an inactive G protein, causing a GTP to displace the GDP. This activates the G protein. The activated G protein dissociates from the receptor, diffuses along the membrane, and then binds to an enzyme, altering the enzyme's shape and activity. Once activated, the enzyme can trigger the next step, leading to a phosphorylation cascade, which ultimately leads to a cellular response, which is in this case a release of calcium ions. Many receptor tyrosine kinases have the structure depicted here. Before the signaling molecule binds, the receptors exist as individual units referred to as monomers. The binding of a signaling molecule causes the two receptor monomers to associate closely with each other, forming a complex known as a dimer in a process called dimerization. Dimerization activates the tyrosine kinase region of each monomer. Each tyrosine kinase adds a phosphate from an ATP molecule to a tyrosine on the tail of the other monomer. The receptor is recognized by relay proteins inside the cell. Each protein binds to a specific tyrosine, undergoing a resulting structural change. Each activated protein triggers a transduction pathway. This protein activates the phospholipase C enzyme that separates PIP2 into IP3 and DAG. This IP3 opens the IP3 gated ion channel, resulting in the release of calcium ions. After insulin enters the bloodstream, it binds to a membrane-spanning glycoprotein receptor. This glycoprotein is embedded in the cellular membrane and has an extracellular receptor domain. Binding of an insulin results in a conformational change which activates tyrosine kinase. This tyrosine kinase activity causes phosphorylation of the enzymes. The two enzymes, mitogen-activated protein kinase, MAP, and PI3K, are responsible for expressing the mitogenic and metabolic functions of insulin, respectively. The activation of MAP kinase leads to the completion of mitogenic functions like cell growth and gene expression. The activation of PI3K leads to crucial metabolic functions such as synthesis of lipids, proteins, and glycogen. In type 1 diabetes, the pancreatic cells that produce insulin signals are lost. Therefore, the insulin signals are also lost. As a result, sugar accumulates to toxic levels in the blood. Without treatment, diabetes can lead to kidney failure, blindness, and heart disease later in life. Some other symptoms are weight loss, extreme tiredness, blurred vision, increased thirst, and many more. There is no known treatments for type 1 diabetes, but it can be managed through various things like healthy eating, regular exercise, and taking insulin shots, which is pretty common among diabetic patients.